So I think what we've seen over the past year has been like a phenomenal global effort, especially from big pharma companies that have put a lot of their resources behind developing these safe and effective vaccines. So it's a Herculean effort, what we've seen in a year. And what it would normally take to generate these years is maybe 30 years of research. So what happened in the past year to generate the vaccines we now have is built on the past decades of having platforms that were ready to go as soon as we knew the sequence of the virus. And I think that really shows the importance of having systems in place to have vaccines that can plug and play for new pathogens. So if we do come across another pandemic within our lifetimes, we're going to be much better equipped to make those vaccines. But what goes into actually making a vaccine is that you first have experimental people like me testing a vaccine in models to see, does it protect animal models against getting infected, against getting sick, against transmission, and what type of immune responses does that stimulate? So you have preclinical research, and the best vaccine that comes out of that then goes to phase 1 clinical trials in humans, and then that is to basically see if your vaccine can stimulate immune response. So what happens in an animal model and a human, sometimes there's a difference of scale and immune response, so you have to check that first works, and that's normally in 10 to 100 people. And then you scale up to phase 2 clinical trials, which is about toxicity and dosage. So you'll start to actually then look at whether it the best type of combination maybe for those vaccines, and that then goes up to hundreds of people. And then you could go up to phase 3 clinical trials, and this is where you actually then look at efficacy. So you'll be looking at 10,000 to 100,000 people that have been vaccinated with either the real vaccine or a placebo, a fake empty vaccine. So you look within those two groups as to who actually got infected, and that's how you calculate the efficacy rate of how protective your vaccine is in the real world, in the real situation encountering the virus to protect against infection. And once we've had recently the emergency use licensure, so this is where the vaccine's benefits outweighs the risks, and this is where we have widespread community vaccinations to fight the virus, and we have post-licensure follow-up. So after a phase 3 efficacy trial, you'll start to vaccinate a wider population, and those vaccines are still monitored for the next few years to make sure that on a larger scale, up to millions of people, that you're still able to observe that there's efficacy against new variants, that there's no unknown side effects. So you go up in scale as that vaccine field progresses, and that's normally done over decades of years. So should we be worried about the fast timescale that these vaccines have been made? I don't think so. I think that the platforms that are being used didn't just come up on day one. So these are actually built on 10, 15 years beforehand of actually doing those preclinical trials. The same vaccine platforms like mRNA and the adenovirus vaccines have already been used in phase 1 and 2 clinical trials. So the backbone of these vaccines has already been tested widespread in a lot of people. And so what we're really just changing is the code that's within those vaccines. So we're using these tried and true platforms. Although they haven't been used at the same scale, they have been studied for many, many years before. And inactivated vaccines have been used for the past 80 years. So it's a way that we take the virus and kill it and then give it to our immune system. So these vaccine platforms have been used for many other viruses. They've been used in the lab in other clinical trials as well. And adjuvants such as alum has been used for over 100 years. So I think that the platforms and technologies that are used in these vaccines is really well known. The vaccines that we're currently using have been through those 3-stage vaccine trials. So they've been tested for safety, for dosage, and for efficacy. So we have safe and effective vaccines.
When we first develop vaccines for a new infection, it's going to be tested on adults first. In fact, the very first trials are usually in men, because if they do it in women and it turns out the woman's pregnant, it's not ideal. Most likely the vaccine's fine, but the worst case would be that there's some problem with the vaccine and there's a mother who's pregnant. So we first do it in adults, and then at a later stage, if it's all okay in adults, then we'll move down to teenagers, and then after that, move down to younger children. So right now, COVID vaccines are being tested in teenagers in different parts of the world, most likely it's going to be fine. There's no reason to think that a vaccine wouldn't work in a 16, 15, 14-year-old the same as it works in adults. And so probably in a couple of months' time, those results will come out. They'll say the BioNTech vaccine, maybe the Sinovac vaccine are fine for teenagers, for adolescents, and then that age group will be invited to get vaccination as well. And we've ordered enough vaccine for everybody in Hong Kong. And then maybe later, maybe not till next year, younger children would also become eligible for vaccination after those trials have been done. But it's always adults first, and then older children, and then finally younger children.
There's a number of vaccine candidates that haven't made it through the preclinical phase or that haven't shown immunogenicity at the phase 1 clinical trials. So we call this the valley of death. So in the clinical development of a vaccine or a drug, 90% of those are actually going to fail. So you're not actually going to end up with widespread population use on everything that you develop in the lab. And failure of a product can happen for a number of reasons. So in Australia, we had a vaccine from the University of Queensland, which was based on folding together that spike so that it retained the conformation. And part of that domain that helped the refolding was based on a HIV protein. So they found that people that had this vaccine in the phase 1 clinical trials then had a HIV type antibody response. Although it had nothing to do with HIV and it helped make a better SARS-CoV-2 vaccine, that vaccine was dropped from further development. So you can have vaccines fail and drop out of the race of development for a number of reasons. And sometimes this can be due to that they don't scale up or they're not as competitive as some of the other products, which is what's so surprising about the SARS-CoV-2 is that we have a handful of vaccines. Like you can pick your flavor, and that's within a year that we have safe and effective vaccines. So I think that that's really promising.